Yeah. Hi, everyone. My name is Hunjun Lee. I'm a PhD student in the biomedical engineering program at the Johns Hopkins University School of Medicine, and I'm currently in Rebecca Schulman's lab. And thank you so much for inviting me to this session. I apologize that this whole presentation is essentially going to be in entirely in English. I'm not the greatest Korean speaker here, especially when it comes to academic presentations. So hopefully you guys can understand. So my research focuses on developing molecular tools for measuring molecules. And measuring molecules is essentially a fundamental challenge in the bioengineering field. And current lab techniques such as ELISA, Western blotting, mass spectrometry, these are all time consuming. They can be laborious. They require specialized instrumentation, facilities, and trained personnel. What's really killer though is they can only measure one molecule at a time, so they cannot be modularly used. So in order to address these problems, molecular biosensors have become of interest. And they can be useful because they can accurately measure the concentrations of proteins and biomarkers, and they can be useful for various research and diagnostic applications. So unlike ELISA, these are designed to be, in theory, one-pot systems, where you can minimize human intervention with minimal washing incubation steps. They can be designed to be used outside of laboratories. They can be designed as portable assays. They can be deployed to different fields. They can be used outside, essentially in different areas and regions. And finally, they can be coupled with materials and interfaces for functional molecular outputs. For example, delivering some sort of drug in response to a cancer marker. And here's the general workflow when it comes to designing molecular biosensors. First, you want to have a binding interaction between the target ligand and a receptor. Then you want to be able to measure this binding response using molecular circuits in order to transduce this. And through this transduction process, you want to be able to produce a molecular output. And then you can usually read this in the form of a fluorescent signal or a colorimetric signal. And various design approaches have been reported in the literature to design these kind of molecular biosensors, usually in the form of antibodies, protein switches, and aptamers. And for the uninformed, aptamers are single-stranded DNA and RNA strands or different kinds of nucleic acid strands that can bind to proteins, small molecules, and different kinds of ligands. The challenge in this field, though, is how do you modularly design these kind of biosensors? Because coupling different protein binding responses with transduction circuits is a major challenge. The question really becomes how do you plug and play different kind of combinations of ligands and binding circuits to molecular circuits downstream? So our lab is focused on this field of DNA nanotechnology, and we use DNA instead of using it as a system, something that encodes a living system, we use it as a material or a structural component when it comes to constructing complex structures such as DNA origami or programming chemical circuits and reaction networks. So really the question for us became, can we couple this broad library of aptamers with DNA and RNA computation to design these programmable reaction cascades and to transduce the binding response of the aptamers? Therefore, our lab developed ARTIST, which stands for Aptamer Regulated Transcription for In Vitro Sensing and Transduction. And ARTIST is designed to be a versatile platform to detect a wide variety of proteins, couple them with different molecular circuits downstream, to design protein biosensors that can have different kinds of biosensing modalities. And we designed ARTIST such that in the absence of a ligand, T7 RNA polymerase, which is actually a well-known bacteriophage enzyme, can actually transcribe this DNA template and produce an RNA output. But when the protein binds to this aptamer, this represses transcription, and you can imagine this as analogous of a DNA transcription factor binding mechanism. We wanted to couple aptamer-regulated transcription then with molecular circuits downstream and use that as a way to construct protein biosensors. So ARTIST has these short DNA templates called DARTs, and they stand for DNA template for aptamer-regulated transcription. And they're composed of a promoter, which T7 RNA polymerase can bind to and initiate transcription. Then you have an aptamer domain that can bind to a specific ligand. And then you have an output domain that encodes some sort of arbitrary sequences to react with molecular circuits downstream. And with that, we designed DARTs to encode this RNA output in this kind of form of a hairpin. And we specifically designed it to be in this form because the complementary sequences of this aptamer are also transcribed in the absence of the ligand. So T7 RNA polymerase would also be able to transcribe this complementary sequences of this aptamer. And this is usually typically very long, and it can also form undesirable secondary structures with this output domain, and that can interfere with its reaction with other nucleic acid circuits downstream. So in order to pretty much isolate this aptamer complementary domain with this output domain, we formed this insulation domain between them that would actually form this kind of hairpin structure and prevent them from interacting with one another. So how does aptamer-regulated transcription work? So when the RNA is produced from the start, it can react with this DNA reporter complex, which is a pre-annealed form of a fluorophore-modified strand and a quencher-modified strand. And the RNA output can then react with this reporter through a reaction called Toho-mediated strand displacement. And to really help out those who are not very familiar with this RNA transcript can actually bind to the single-stranded region here. It's called this overhang called the toehold on the DNA reporters. And when it does, it's competing with this incumbent fluorophore-modified strand through a process called branch migration. So this fluorophore and this RNA are sort of competing with one another on this quencher-modified strand. But because this RNA is fully complementary to this quencher domain, it's going to pretty much knock off this fluorophore-modified strand. And we can actually measure the fluorescent signal coming out of this released DNA strand, and we term this the reactive reporter. And this is happening over time, so we sort of refer to that also as reactive reporter kinetics. And because we can calibrate this to some sort of concentration, this initial reporter concentration we added into our assay, then we can have it as a function of concentration. But all this to say, however, when the protein is actually bound to the start, then there is no reporter reaction, so it will remain unreacted because transcription is repressed. So just to help out with a little bit of nomenclature, we first designed a DART that has an input that has an aptamer domain for interferon gamma. It's based on this aptamer, and then you have an output domain that is encoded downstream of this aptamer domain. So basically, we term this DART based on the input and the output domains of this template, IFN01 DART. And we first subject this IFN01 DART to different concentrations of IFN gamma across 0 to 1,000 nanomolars. And what you can notice is with increasing concentrations, you see this decrease in the reactive reporter kinetics. So we asked, can we model this binding between this DART and IFN gamma based on the differences in this reactive reporter kinetics? So to have a better understanding of this, we designed this quantitative model that can help us predict the binding affinity of DARTs. And this is based on this dissociation constant, this KD value, which is basically the ratio between the concentration of the ligand, protein ligand, and the concentration of the unbound DARTs over the concentration of this complex between the protein ligand and the DART, all under equilibrium. And if we expand this and reorganize these equations, then we can basically have a function of the concentration of the ligand-bound DARTs as a function of the concentration of the initial concentration of the DART, the ligand, and the KD of this aptamer, all under equilibrium. And based on this, we can now calculate the concentration of the DARTs that are unbound, because we're assuming that when it's unbound, then it can produce RNA. We can feed this now into a mass action kinetics to calculate the concentration of RNA that's being produced over time. And then based on this, we can also calculate the concentration of the reporters being reacted over time. So we measured the dose response curve of IFN01 DART across 0 to 1,000 nanomolars, which is actually the reactive reporter concentration at 120 minutes across 0 to 1,000 nanomoles of interferon gamma. And then we were able to fit this KD apparent to this dose response at around 8 nanomolars. And then you can also see that the kinetic plots, the model and the simulation and the experiments seem to match pretty well with one another. The next question became, can we plug and play different protein binding aptamers into DARTs? So we found 4 different aptamers that can bind to different proteins in the literature. One that binds to interferon gamma, thrombin, interleukin 6, and TNF alpha. We subjected each of these DARTs across 0 to 1,000 nanomolars to their corresponding ligands. And you can see there's a similar trend where there's an increase in the ligand concentration results in a decrease in the reactive reporter kinetics. And this was consistent all throughout. The next question became, are they selective towards a specific to their specific ligands? Will they only repress transcription only in the presence of their specific ligand? So we took each of these darts and subjected these across this library of proteins. And you can see only in the presence of its corresponding protein, there's a decrease in the reactive reporter concentration. And as a control, we have this dummy dart, which has pretty much an aptamer domain that doesn't bind to any of these proteins. And you can see for this one, there is no decrease in the reactive reporter concentration across this panel of proteins. Instead of changing the input domain, this aptamer domain, we asked, can we swap out this output domain? We keep this IFN dart, but can we encode different arbitrary sequences for the output? So we designed 3 different darts, IFN01 dart, IFN02 dart, IFN03 dart, subjected them across the same range of IFN gamma concentrations, and you can see there's a similar trend where with increasing concentrations, there's a decrease in the reactive reporter kinetics. So now that we've sort of explored aptamer regulated transcription, and we've seen the modularity of designing these different darts using our system, we asked, can we use this as a protein detection layer, couple this with molecular circuits downstream to construct different kinds of biosensors. And we largely developed 2 different kinds of sensors. One is an analog sensor. And this basically follows the same kind of mechanism you saw with aptamer regulated transcription with the darts alone. But what we have instead is once the RNA is produced and reacts with a reporter through Toho-mediated strand displacement reaction, we also include RNase H. And what RNase H specifically does is it targets this RNA that's bound to this quencher-modified strand. It's specifically targeting the RNA that's bound to a DNA complex. And what's going to do is it's going to degrade this RNA, and then now this quencher-modified strand is going to be single-stranded. This can actually recognize a single-stranded fluorophore-modified strand, quickly rehybridize, recreating this unreacted complex. So what you essentially have is you have production of RNA reacting with the reporter, as well as degradation of RNA that's unreacting this, basically creating this unreacted complex. So this steady state production and degradation is giving you these steady state outputs. So we brought back these 4 darts we characterized, and we try to convert them into analog sensors. And you can see all of them are reaching steady state at around 120 minutes. And this is very different from that initial dart that I've shown, because those were basically the concentration of the ligand is reflecting the transcription rates. But here, the concentration of the ligand is reflective of these distinct outputs, these steady state outputs of each of these different sensors. The next construct we developed was one that produces a digital signal. And this is basically designed such that below some sort of concentration of protein, you will get a low signal. Whereas if you have a high concentration of protein above some sort of threshold, that's going to produce a very high signal. And this is composed of a comparator circuit. And it's basically comparing the transcription rate of 2 darts. You have a reference dart here that's producing reference RNA. And this reference RNA can react with a reporter through Toho-mediated strand displacement. You also have this other dart here that has an aptamer domain that binds to a specific protein, interferon gamma in this case. And this is encoding some sort of RNA that has a complementary domain to this reference RNA. And this reaction actually occurs really quickly. This is a thresholding reaction. It's a hybridization, so it's almost an order of magnitude quicker than this reporter reaction. Therefore, what we can essentially have is below some concentration, we have a very low output because a lot more of this IFN01 prime dart is producing RNA. Whereas if you are adding sufficient amount of IFN gamma to suppress this, then you have more of this reference RNA being produced, being able to react with this reporter. So using simulations, we first tried to design this digital sensor that turns on above 50 nanomoles of interferon gamma, but is off below that. And we were able to use experiments to pretty much validate this on-off digital response. We then asked, can we tune the detection threshold of these digital sensors by simply changing the reference dart concentrations? And what we realized is that if you increase the concentration of this reference, then you can allow this digital response to occur at a lower concentration of interferon gamma. Whereas if you decrease the concentration of this reference dart, then you can have it respond at a higher concentration of interferon gamma. We next evaluated robustness of these digital sensors because these are supposed to only create, on or off, 0 or 1 kind of, signals. And we asked, for different environmental assay conditions, would that, affect this kind of on-off response? So we looked into T7 RNA polymerase activity, this enzymatic activity, as well as the different ion concentrations within the samples. For example, like, serum or urine or different bioass samples could have different ion concentrations. So we try to see if that could affect the response of our sensors. So we first try to simulate with different transcription rates how that would affect this digital response. And then we try to use experiments using different concentrations of T7 RNA polymerase, how that would cause effects on our digital response. And you can see all of them have a high output at 50 nanomoles of above 50 nanomoles of interferon gamma, whereas below that you have a low output. And then this digital response seems consistent throughout. And then we try to see how does the digital response change with changing concentrations of potassium ions. And we designed these digital sensors using 2 darts that are similar sensitive to potassium ions because both of them have this kind of G quadruplex structures. And we postulated that this would cause them to equally change to different concentrations of potassium. And you can see with this kind of calibration effect, they all produce a digital response regardless of this potassium concentration. But if you have the reference not being able to form this G quadruplex, this essentially breaks this comparator and it will not function properly at low concentrations of potassium. So up to here, we've shown that we can actually tune the detection threshold digital biosensors by simply changing this reference dart concentration. So narrowly changing the detection threshold. But we asked if we can make our biosensors even more sensitive. So we tried to see if we could couple digital sensors to another class of in vitro transcription templates, and that can act for amplification. And we call these GLITs in our lab, and these are short transcription templates that can be either on or off based on the binding and unbinding of DNA and RNA components. And I'll talk a little bit more in detail when we go further into the slides. But basically, their on rates and off rates will determine whether or not RNA will be produced by these templates. And if you can almost think of these templates as some sort of like multiplication function. So what we try to do is once you have a comparator producing some sort of on response, could this be used to activate this gene? So essentially initially these GLITs are off. These have an incomplete promoter, so T7 RNA polymerase cannot bind to it, initiate transcription. And this is actually further prevented because there's a DNA strand here that's actually preventing activation of this GLIT. But what the on response of a comparator would do, it's going to bind to this strand here. It's going to bind to it. It's going to turn this GLIT into an intermediate phase. And once it's in this intermediate phase, we have these free-floating DNA activator strands that can recognize this toehold on this promoter region. It can quickly bind to this promoter. It can activate this GLIT, allow this GLIT to turn on and produce RNA. So once it's being transcribed, we can also measure the fluorescence. We can measure the concentration as well as the fluorescence of this RNA reacting with the reporter. One other thing to mention, though, is that we also have RNase H here. And what this does is it's actually acting as like a degradation component. It's actually allowing this blocker to be single-stranded. And once this is single-stranded, it can actually bind back to this GLIT on its on state or either in this on state or this intermediate phase, recreating this blocked complex. So this is sort of also another thresholding kind of effect. So using this amplification scheme, now we were able to tune the detection threshold down to 1 nanomolar of interferon gamma. And this is great because now we've shown that we can also we've shown tunability by 2 orders of magnitude. So really all this to say, ARTIS is a powerful systems chemistry approach to couple the binding response of aptamers with molecular circuits downstream. And currently we're interested in integrating ARTIS into materials because the Schulman lab does a lot of work with hydrogels too. And we're trying to look into more practical applications. For example, in tissue engineering or in sort of cell sorting applications, or even having ways of sensing proteins over time, producing some sort of functional outputs in response to these kind of proteins. So that's all I have for today. I would like to thank my mentor, Rebecca Schulman, as well as Sam Shafter, who is currently a staff scientist at NIST. Also lab members, including Byung-Hwa, Dong Wu, Yan, and all other people who have also contributed to my research. Also would like to thank the research grants, NIH, NSF, and also my fellowship, the Aspen Foundation Fellowship. And thank you for listening.